Welcome to iCANX. We're delighted to have you with us. I'm Paul Weiss from UCLA, and yes, there we go. I'm on the faculty here in nanoscience, and we're delighted to introduce the November program. Today we'll have Professor Trujillo-de Santiago. Coming up, we have Lan Yang, Yuri Vandenberg, and Hong Bo Sun. So please stay tuned and stay with us for the rest of the month. And next slide. So I'll serve as the moderator today. We're hoping that our co-lead, Professor Martin Tuo, will be able to join us from Kenya, where he is today, and we'll be joined also with our founder, Professor Alice Zhang, and I think you all know Alice. I'll give Professor Alvarez's introduction in just a moment. But the main feature today is Professor Grissel Trujillo de Santiago. She's on the faculty of Tecnologico de Monterrey and is the Chief Scientific Officer of Forma Foods. Her research focuses on the development of technologies based in chaos to produce multi-layered and multi-layered 3D microstructured living tissues. She's a member of the National System of Researchers in Mexico, Level 2. She's an associate editor at ACS Materials Letters, one of our favorite journals, and a scientific advisor for the United Nations University LATAM. Her many awards include the Young Researchers Prize from the Mexican Academy of Sciences, the Fellowship for Women in Science from L'Oreal UNESCO, the Romulo Garza Prize in Entrepreneurship, and being named a Distinguished Citizen by her hometown, Matamoros, Coahuila. She's a member of the Distinguished Circle of Faculty Members of the School of Engineering and Sciences at Tecnologico de Monterrey. She's a terrific scientist, engineer, mentor, colleague, collaborator, and friend. And let me pass the floor to Professor Trujillo de Santiago. Welcome.
Thank you. Thank you so much, Paul and Alice, for that lovely introduction. And thank you for the opportunity to present our work in Mexico here today in this wonderful forum. So just let me put my slides here so we can start. Thank you. Thank you so much. I was telling Alice that it's my dream to come to China, to visit China. So hopefully sometime soon we have the opportunity, but at least my voice and what we do, it's going to be in China today. So I'm so happy for that. So, well, today I'm going to talk to you about chaotic bioprinting. This is a technology that we invented in our university in Tecnologico de Monterrey, together with Dr. Mario Alvarez. So to start, let me present you our group. This is the Alvarez Trujillo lab. It's just the combination of our last names, Alvarez, Mario Alvarez and Trujillo. And well, this is our team, many graduate students, undergrads, a few postdocs, and well, very passionate people wanting to change the world. And among our dreams is to have the ability of sometime be able to fabricate full functional organs for patients that need them. Of course, there are many things to do. We are still not there yet, but I think we are advancing in this field to actually make this dream come true in some decades. And many great advances are happening. These are some technologies that allow us to do several things. For example, producing multi-material constructs using hydrogels and cells, living cells. Also, people have developed amazing technologies to make volumetric living constructs with hydrogels and cells within seconds. Large structures with no restrictions, basically, in the terms of form, and also highly dense structures with perfusible systems, and just to name a few. But, you know, I think there are no only one strategy or only one technology that is going to bring us to this stage in which we are actually capable of producing full functional and complex tissues and organs. And there are many challenges to overcome. And we as a group, as the Álvarez Trujillo Lab, we decided to focus in these two key characteristics of our tissues. The fact that they are microarchitected and that they are composed of multiple materials. And if you see histology images of our tissues in our whole body, you will find that there are many types of cells and in our tissues arranged in a very special way. They are not just homogeneously put there in our tissues. They come with very fine structure at the micro scale. So that's why we are very interested in this microarchitected characteristic of our tissues. And well, we came with this idea many years ago that chaotic flows could help us to fabricate these structures with these characteristics. And well, that's where the chaotic bioprinting idea came from. And well, in chaotic bioprinting, we use a suspension of cells, like imagine that this will be the suspension of cells with this color fluid. And then we make these structures with flow within a hydrogel precursor while it is still liquid. And then we cross-link that hydrogel with the structure of the cells to preserve the structure that was drawn by the movement of the fluid with the cell suspension. So this is basically how chaotic bioprinting works, but we use a very special type of flows. These are the chaotic flows. Because with the example that I just showed you, it will be very difficult to always reproduce the same structure. And we can achieve the same structure and in a reproducible way using chaotic flows. And here I want to be very careful and say that chaotic flows in this context have nothing to do with disorder or turbulence. This is the deterministic chaos, the mathematical chaos, the one that I'm talking about. So just to try to forget a little bit about that connotation that we use for chaotic flows as a disordered process. And well, let me tell you a little bit about the story on how we came up with this idea. And it was when I was looking at the research that Mario made during his PhD. I learned from him that chaos is used to mix very efficiently in the laminar regime. This is in the context of chemical engineering, to mix big bioreactors or big reactors for the chemistry industry. And people in the chemical engineering knew since the 90s that chaos produced these layered beautiful structures along the process of homogenizing viscous liquids. But nobody was using this ability of chaos to structure materials until, well, we realized that we can use these structures to make cell-laden hydrogels with these structures made by chaos, using very benign conditions, because this happened in the laminar regime under very benign fluid regime, very slowly. So some characteristics of chaos that we are taking advantage of is that, again, they are deterministic. I already explained that they can make a structure. And well, since they are deterministic, they are also reproducible and predictable. Actually, what you can see in the screen is the outcome of an experimental experiment of chaotic printing, the one right here made by Carlos and Joanna, and then the computational simulation that Diego made. So you can see that the structures matches. So we we can do this. If we repeat the process, it's going to have always the same outcome. And very importantly, they make a structure in an exponential manner. So I'm going to explain that later. But that is another characteristic that we are using from chaos, that they are very efficient at making a structure. And very fortunately, these layered structures that chaos can make are very, very common in our tissues. Our tissues are very often made by layers of cells sharing interface with a layer of another type of cell or another type of material. So somehow this is a signature of nature that we find in our tissues. Even within our cells, we can find that, for example, the endoplasmic reticulum is made of nanolayers, right? So why not using this ability of chaos of making layers to make engineered tissues? And well, let me quickly show you our most simple setup. This is the chaotic printing head. We call this the iCANX static mixer. This was have been used for a long time for mixing very viscous liquids. And basically what we do is we use very short static mixers to preserve the structure. So they have this ability to make layers, intercalated layers, but we don't use very large static mixers to homogenize, but short mixers to make a structure. And this static mixer is composed of several mixing elements. As you can see here, this is the static mixer that comes inside this pipe. They actually are static. They don't rotate, but here I'm just showing you the rotation for you to see that every mixing element is exactly the same, but they are positioned 90 degrees with respect to each other. And what they do is that when you perfuse a liquid through them, let's say this is a hydrogel, the hydrogel streams get reoriented and then split in every mixing element, and forming these intercalated layers that you can see in this cross section. So here we are using alginate. The alginate crosslinks when it comes in contact with calcium chloride, with divalent ions. And so you can extrude meters, literally meters of this hydrogel fiber. And if you cut, you will see this structure along the entire filament, exactly the same structure. And as you can see here, we can model this, we can make computational simulations just to show that the outcome is the same experimentally and computationally. And you can decide how many layers you want, depending on how many mixing elements you use in the printhead. For example, if I use only one, what you will have, it's only 2 layers. If you use 2 mixing elements, 4 layers, with 3 mixing elements, 4, with 4, 16, with 5, 34, and so on and so forth. So with this strategy, you can multiply by 2 the number of layers that you have in the previous section in a very easy way, just by adding 1 more mixing element. And well, this is amazing for us because, for example, if you are using living cells, the mammalian cells are around 10 microns in diameter. So if we use this configuration of the iCANX static mixer with 6 mixing elements, you will have 64 layers inside your construct, inside a filament of 1 millimeter diameter, and with lines of single cell width. And so this is amazing because so far the extrusion technologies of bioprinting out there cannot reach this resolution. And that's why this is because if you want to achieve this resolution of 15 microns or 10 microns, you will need to use a very narrow nozzle. And this is very difficult to do in bioprinting because if you use very narrow nozzles, you are going to kill the cells. Basically, the shear stresses are going to increase a lot, and it's not a very good idea to use very narrow nozzles if you want to print cells. In chaotic printing, the resolution is not dictated by the nozzle, the nozzle size, but by the number of mixing elements that you are using. And the outlet can be huge. It's 1 millimeter in diameter, so the cells don't even notice that they are being extruded through this printing head. Well, this is not only across, this is what I'm showing is a cross-section, but remember that this is a cross-section of a long filament. So we have this structure within the whole construct, which is also something that we find very useful for bioprinting. And well, other thing is that as I mentioned, the narrow nozzles are our enemies in bioprinting. The narrower the diameter, the more shear stress, and the more shear stress, more cellular damage. In chaotic printing, we can get very small features. The resolution, meaning the size of the layers, can be very small, but we use outlets way larger, around 1 millimeter. So cells feel happier that way when they are extruded through large nozzles. And to prove this, we printed murine myoblasts, in this case C2C12 cells. These are live and dead stainings, when you can see the living cells in green and the dead cells in red. And well, we observe very high viabilities, higher than 85% in our constructs for 28 days of culture. And then this makes sense, again, because the outer diameter is huge. So cells are being accommodated by the laminar flow very benignly. And well, this iCANX static mixer in particular, it doesn't generate very high drop pressures. There is drop pressure, but it's not unsuitable or aggressive for the cells. In the shear stress map, you can see that indeed there is a profile of shear stress within the static mixer. You can notice that there are few red spots that could damage the cells, but most of the flow passing through the static mixer is not subjected to high shear forces. While narrow nozzles, as you can see here in this comparison, definitely is going to be very high, and it comes with other limitations like the throughput, drop pressure, and limited resolution. So far, I have shown that cells are living, but it's also important that they are functional, because at the end of the extrusion, they can be alive and kicking, but we don't know if they are going to be capable of actually making tissue. So for us, it's important that they are capable of making tissue. So cells not only survive, but they also show signs of tissue maturation. This is, again, the cells, the precursor of skeletal muscle, C2C12 cells. And at the beginning, they are immature cells, like in this cartoon. They are myoblasts. But if over time, if the conditions of the bioprinting process and the culture conditions are favorable, they are going to by themselves evolve into these myotubes, more mature cells of functional skeletal muscle tissue, and they will express this sarcomeric alpha-actin that is present in these myofibrils. So here what we did was this immunostaining that is going to reveal if that sarcomeric alpha-actin is there or not. And what you can see in red, that fluoresces in red, is that sarcomeric alpha-actin stained. So it was present, and we were happy because after some days of culture, that protein that can let us know if the cells were functional is there. Of course, this is still not perfect, mature and functional skeletal muscle tissue, but this is a sign of maturation of the cells. Although there is a problem here, because we realized that the cells were growing only in the surface of the construct. This is a cartoon showing you like more or less how was the filament, and that layer of cells that you are looking at here, they were aligned and kicking and happy and forming something similar to skeletal muscle tissue, but they were only in the surface of this big construct of 1 millimeter thickness. And of course, this was not a surprise, because there is a limitation of the mass and oxygen transfer in thick hydrogels. And actually, this is a big challenge in tissue engineering, that is not so easy to perfuse nutrients and oxygen to thick hydrogels. So this is, we have this problem, and we wanted to solve it. And well, together with Professor Paul Weiss and Ali Kadem-Hosseini, we came up with this idea of using also chaotic printing to make vessels, to make empty channels within our constructs. So it occurred to us to use a sacrificial material. So use a permanent material that is going to form the filament of hydrogel, and a sacrificial material that is going to remain liquid, and it's going to be removed from the construct, from the fiber, and it's going to leave empty channels that can act or can work as blood capillaries. So it worked out. With this strategy, we can get hydrogel filaments 1 millimeter thick. Again, big for cells. This is a huge dimension for cells, but containing perfusable hollow channels inside that can help us to bring the nutrients, to remove the byproducts, and also to incorporate oxygen for the cells that are living inside our hydrogels. So, well, here I'm showing you that we can perfuse actually the liquid through the hydrogels, but if we don't perfuse the liquid anyway, we are going to increase the mass transfer just by diffusion. And I'm going to activate my video here. So what we have here is a fitzy-dextran inside the hydrogel. And as time advances, you can see that the fluorescence decreases because it's diffusing out from the filament. So when we have channels, which is in this case and in this case, the fitzy-dextran or the fluorescence, it's getting out from the filament faster because there is more empty space. We have more permeable or diffusible, if you allow me the expression, material than in the solid one. In the solid one, the fitzy-dextran stayed within the hydrogel. So we were happy with this, and Carlos and Joanna, they are very ambitious, and they want to push the technology forward, and they were capable of having some degree of control in our process to produce more or less channels, empty channels within our hydrogels. So we were capable, this is a beautiful image, I found it beautiful, where we can, they showed that they can incorporate up to 31 channels inside a 1 millimeter filament of hydrogel. So this is amazing. It's very difficult to get so many empty channels that are going through the whole filament with hydrogens. So with this, we were even capable of getting hydrogels channels as narrow as 20 microns. This is already the dimension of a blood capillary, which is not trivial to get with other techniques. So we were really excited about this, to have this high density of hollow channels within a soft material such as a hydrogel, because, well, we need that extent of dense microvascularization in our body if we are expected to make thick hydrogels or tissues in the future. So next, to prove the concept that this is prevascularized, we need to incorporate cells. And again, we used myoblasts, C2, C12 cells that were laden in the permanent hydrogel. And then we use a sacrificial material, in this case, hydroxyethyl cellulose. And we observed several things, as you can see here. In the filaments where we introduced the channels, the cells were spreading more than in the ones that they didn't have channels. And also the cells at day 7, they exhibited a higher metabolic activity and higher cell viability. This what you can see in these two graphs, than in the ones when they were present in the filaments with channels than in the solid filaments. And also we look for markers of hypoxia and proliferation, and the cells that were present in the filaments with channels, they exhibited lower expression of hypoxia markers and higher expression of proliferation markers in comparison to the cells in the solid channels, as expected, because they were they had access to oxygen and nutrients, and they were happier in these filaments. And well, in the filaments with channels, also the cells they merge together. What these cells do, let me show you here. What cells do when they are happy, these myoblasts or these precursors of skeletal muscle tissue, they start to spread and they merge together, forming multinucleated cells that we call myotubes. So what we saw in our constructs with channels, it is this transition from myoblasts, the immature cells, to myotube, the mature cells. And also this had relationship with the markers that they were expressing over time, as we reveal with PCR experiments. Also, the cells that were printed with the channel filaments, they were longer because they merged more efficiently, and they had more nuclei per myotube or per cell, as expected. And very nicely, they were also more oriented towards the direction of the main direction of the filament. And as you can see here, the solid ones, they were all over the place, but the ones that were printed in the channels, there were like soldiers very well aligned towards the main direction of the channel, because actually the channels gives them that physical cue. So you have to attach to the surface and move that way and merge with your next cell neighbor to make a strong and solid myotube. So we saw what we expected, happily. And well, we have these filaments with channels, right? So why don't we use those channels to grow cells inside them? Because of course it makes sense to use them as prevascularized tissues, but we can also use the channels to grow tissue inside those channels. And this is what we are doing with Carlos Ceballos. He's growing cancer microtissues inside the channels. Actually, this happened naturally. We didn't intend to do that. They were at the beginning in the permanent ink, but these cancer cells that he's using are very mobile, and they are very prone to invade other environments. So they started to invade the channels, and they form these beautiful microstructures, very dense microstructures inside the channels. So now we are thinking, why not using this as a template to grow other type of tissues? In this case, is cancer cells as to study cancer as a model, but we can also translate this to dense tissues like muscle or other tissues that you might be interested. So that's what we are working on right now. And other thing that we can take advantage of chaotic printing is that we can make compartments within the whole volume. And we have this phrase in Mexico. I don't know if this makes sense for you guys, but we say juntos pero no revueltos. That means together but not scrambled. So sometimes we want to have cells that are working together very close to each other, but we don't want them scrambled. So this can help us to make structures with them. For example, in this case, we alternated inks that were responsive to cells and other inks that were not responsive to cells. So the cells, they cannot cross to the barriers of the non-responsive cells. and they are obligated to align in the compartment in which they were printed initially. As you can see here, this was scrambled, this was premixed, so we cannot see any function or structure. And here is exactly the same composition, but we had control over the microarchitecture, and they are aligning as we were we expected. So also there are, in a very similar way, we can use this ability to compartmentalize to bioprint with nanoparticles. For example, bioactive glass nanoparticles. This material releases ions that can be useful to activate or promote certain cell processes, but bioglass in particular is very, very active, so it can be aggressive to cells. That's why we wanted to have them close to cells, but not scramble with cells. So we printed fibers with intercalated layers of cells and bioglass. So it was cell bioglass, cell bioglass, cell bioglass in separate layers. And in this way, we could see that the cells were oriented, and they were happy, and they were aligning, forming this skeletal muscle-like tissue without any side effect of the aggressive action of bioglass if they are very close to bioglass. So again, we saw the expression of the markers that we expected to see over the time, and this was a successful way of printing myoblast with, this case, a nanoparticle bioglass and cells to make skeletal muscle-like tissues. And well, I'm going to bring back this beautiful fiber that I love. And of course, this is a very nice way to incorporate microchannels inside, but there was a problem with this fiber. And the problem is that they are very weak, right? So we are very effective in incorporating hollow channels or microchannels inside, but the compromise is that they are very weak. Actually, we could sustain with these particular fibers only 7 days of culture. So we wanted to have more room for experimentation and to bring these experiments for longer time. And so we needed to reinforce these fibers. And for this, what we did was to adapt our iCANX static mixer, our chaotic printing head, with more inlets or to habilitate to perfuse more materials. So some of these materials could help us to reinforce the cells. So for example, here we could incorporate 8 different materials inside. In this case, you can see 4 different materials that, depending on how you accommodate the hydrogels, they are going to have certain patterns that are going to respect certain positions. You can have control on that. So, but let's say if you have this configuration, we can extrude some, through some inlet ports, hollow sacrificial materials to make hollow channels. And in some others, we can use a reinforcement hydrogel that is going to make our fiber more stable for extended culture extended culture times. And the rest of the materials, you can use whatever you want. It can be cells, different types of cells, or different type of materials that are going to interact with cells. So with this more degrees of freedom, what Andrea did was to explore different configurations and ratios of the bio ink, the sacrificial ink, and the support ink, to see what could be a sweet spot between reinforcing the fibers and maximizing the volume of the cells that we are going to print in our filaments. So what she found out is that these 2 configurations could be a good candidates for incorporating cells. The cells, they don't like very stiff hydrogels. They need room, they need the space to actually interact and to spread and to have interaction with neighbor cells. So that's why sometimes these hydrogels might be very weak, but some others, like the support portion, can be a stiff hydrogel because it's not going to contain cells. So in this case, we can increase the mechanical properties while still having room for incorporating cells in soft hydrogels. And we also have room to incorporate a sacrificial material that is going to leave the hollow microchannels inside. So using this reinforcement strategy, we were capable of extending our culture times up to 21 days so far. So this is what David Hernandez did with this reinforcement approach. And so before we had only the ability to grow our filaments up to 7 days, but now we can do it for 21 days so far. Of course, we're envisioning more things to happen with this system. We think that is very versatile, very easy to use, and also very cost effective. So, for example, we are thinking right now, we are making this in the lab to incorporate not only the hollow channels, but also endothelial cells that can make this nice endothelial tissue inside, lining our microchannels to make more functional vasculature as we have in our tissues. So to finish, let me tell you again, this is a cost-effective technology. It's very low investment. We are very attractive methods in terms of resolution and versatility and cost. And well, you know, this coming from Mexico, that is a developing country, this aspect is very important to us because we need to make relevant science, but with little money. So it's also a way for us to democratize biofabrication in countries like Latin America. And well, as a summary, chaotic bioprinting uses chaotic advection to create microstructure. It produces the structure exponentially, is a deterministic process, therefore it's predictable and reproducible. It's cell-friendly because we saw not only viability of our cells, but also signs of maturation towards the formation of skeletal muscle tissue. It's high throughput. We can literally print 1 meter of these structured hydrogels with only 1 minute, and we can produce hollow channels as narrow as a blood capillary, and it's a versatile tool. Right now, we are actually using this tool for making meat, plant-based meat, and also cell culture meat in Informa Foods, that is our startup. And well, I close acknowledging my students who are really the ones that make this, all these results a reality, my mentors and collaborators, and the institution and my funding sources. Thank you very much. Thank you for your attention, and very happy to interact with my colleagues in the panel.
Thank you so much for the lovely talk.
Thank you, Paul.
Now, let me bring out our panel. So our first panelist, you heard a little bit about during the talk. Professor Mario Alvarez is a Romulo Garza Insignia Professor at Centro de Biotecnologia, FEMSA, and the Mechatronics and Electrical Engineering departments at Tecnologico de Monterrey in Monterrey, Mexico. He's also a member of the Sistema Nacional de Investigadores, where he has the highest ranking, level 3, and is a permanent member of the Mexican Academy of Sciences. He's a founder and the chief technology officer of Forma Foods, a startup dedicated to the design and fabrication of plant-based meat analogs and cultured meat, which we just heard about. He previously worked at the Pharmaceutical Institute at Bristol Myers Squibb and was a visiting professor at MIT, where he and Professor Trujillo-de Santiago and I first met. His research and collaboration with our speaker combines biotechnology and bioengineering, including biofabrication, bioreactor engineering, biomaterials engineering, mixing, and microfluidics. At Tecnologico de Monterrey, Professor Alvarez was the founder and first director of the Centro de Biotecnologia, FEMSA, and the founder of the PhD program in biotechnology. He is also a wonderful, kind, intelligent scientist and engineer, mentor, collaborator, and friend. Welcome, Mario. Yeah, and then I'm not sure if we have Professor Tuo from North Carolina State University joining us. I hope so. He was looking for an internet connection, the last we knew. And we have, of course, our founder, Professor Alice Zhang from Beida, here with us for the panel.
Okay, great. Yeah. So Paul, maybe first I came to the question. I was so excited, for Grissel's talk. Yeah, it's really exciting. So Grissel acts a fantastic job. Okay, the genius idea, to use a choose bioprintings. So have a question is so now how can layers, right? That depends on how many nozzles you use, right? How many elements. So how about the thickness? Can you control that?
The signals.
No, the thickness.
The
So,
thickness.
The thickness. Yes, yes.
Yeah.
Yeah, there are some restrictions, especially depending on the material that we use. For example, right now we have used a very easy to use hydrogel alginate because it cross links with the calcium. So in the case of alginate, just to give you an idea, we have found that the best size of the outlet of the nozzle will be 1 millimeter or smaller. Because if you use very wide outlets, the cross-linking is not going to be effective in the center of the hydrogel. So it's going to remain liquid. If you want that, that's perfect. In that case, you will have a hydrogel with a big hollow channel inside, right? But yeah, you can control that, and it's going to depend on the material, the cross-linking method, and of course, we want to preserve the viability of cells. So our advice will be to use wide nozzles so we don't have very high shear stress that can affect the cells.
Okay, yeah, this was a super. How about another question is I see that, the demonstration is quite simple, as very cheap. So, of course, welcome this. How about questions? Looks like these simple devices, you do control the temperature and other parameters, right?
Yeah. Also, alginate, in the case of alginate, it's super easy to work at room temperature because it's not going to change phase from liquid to solid at room temperature, let's say 20 degrees. But for other hydrogels like gelatin or collagen that are also very frequent types of hydrogels for bioinks, they do have a more tricky behavior in terms of temperature. In that case, yes, the temperature should be very well controlled. Otherwise, it's going to be a mess, and we are not going to preserve the structure. Yeah.
So, Paul, can I ask a question to Professor Merrill? Yeah.
Yes, actually, related question. How much can you control the porosity? Important for growth, and then I have a follow-up after that. Yeah.
Yeah.
Yes,
Please, Mari.
yes, the porosity, that is actually, Paul, one of our aims, to be able to very finely control the porosity. Because right now we are exploring diverse bio applications in which sometimes we need sufficient space for small cells, like mammalian cells, 10 microns in diameter. But sometimes we want space for spheroids. And in that case, we are aiming for holes or voids in the order of hundreds of microns and even 1 millimeter. So what we are doing now to control the porosity is two things. If you change the number of mixing elements, then you will go for more layers. And then since the diameter at the outlet is fixed, you divide that diameter in more layers. So the average thickness is smaller. And the other way around, you decrease the number of elements, you have wider striations. But there is another clever way that Grissel found to control the porosity in a different way. If we go with inlets in the top, all the striations that you end up with are more or less have more or less the same thickness at the outlet, at least in the central line of the fiber. But if you enter with lateral ports at different stages of the mixer, let's say you have inlets, lateral inlets at the beginning or at the end, the liquid that comes in those inlets has less elements that will divide it. So that's a very effective way to control the thicknesses of the materials that you inject in those lateral inlets. So let's say that we want wider voids. So we can inject a fugitive ink at the end of the mixer. So we will have less divisions of the fugitive and as a consequence, wider voids in the fiber.
Mhm.
So in some, there are clever ways in which you can make a little bit more heterogeneous the distribution of the thicknesses of the layers. And as a consequence, you can control the average diameter of the porosity that you have in the fiber.
Very good. So actually, a related question, what about surface functionalization? It's something, I would imagine you probably have to do as you do the growth. I think this paper where we did grew the gold nanostars in the microfluidic channels. It took a fair bit to make that happen uniformly. So you're better off, especially, if you're going to make these long structures to do it as you grow. Do you have some thoughts in how you might manage that, and that also may be a way to to control the growth or enhance or even package organoids or spheroids or something else that you might want within?
Well, I love that idea. Oh, it's amazing. And I think that there are many things that we can do. So far, again, we have used very basic systems. It's just alginate and Gelma. But we are eager to explore more things. And I think we need the collaboration of chemists like you, because we need to find also a way to functionalize more our striations inside, so to provide a better environment for cells. Because as in our tissues, it's a structure, but it's also physics, but it's also chemistry, and everything interplay in all those factors merging together, right? So yeah, I sometimes when I dream and we dream together, Mario and I say I would like to have like a magical material that can come and then make like the membrane of the cell inside these filament, like very long membrane layers with proteins in there, like having interfaces that are really functional for cells, not only a basic hydrogel, but something more similar to our own cells and tissues. So I think so far we don't have those tools that developed in our lab, but we can't wait to start adding more, not complexity just because complexity, but functionality to this structure. Yeah.
Well, you have to
May
send
I?
the star students like Joanna and Carlos up to Los Angeles so we can work on it together. Mario, you were going to say something.
Yes, Paul, yes. That's a beautiful question. And let me share that we are one idea in which we are starting to work is functionalization with different enzymes in different channels. And the first approach to this has been immobilizing enzymes in nanoparticles. And then we feed these nanoparticles in some of the streams. So that is a very effective way because the nanoparticles will not diffuse through the hydrogels once they are cross-linked. So the mobility of these nanoparticles is basically negligible. So you have functional layers now And we have different ideas to you see the basic idea is what I described, but then we are exploring different scenarios to illustrate the potential uses of this idea. One that is interesting, and we're exploring that with a colleague in the food department here in Monterrey Tech, is to induce flavors. So imagine that you have a fiber and you have these different enzymes and you have these different ingredients in other layers.
Mhm.
So when you break the fiber, let's say that you have an eatable fiber, then you are going to expose the substrates in some layers to the enzymes in other layers, and you will release flavors that otherwise you will not feel if you don't break if you don't chew the fiber. So that's just an example of materialization of your idea, right? How making these different layers functional and segregated can uncover applications that are kind of interesting in different fields.
Yeah,
So,
that's very nice.
fantastic.
Actually, I'm still thinking if that we can print, liquid metal inside. Yeah, can get more function. Yeah, because
Mhm.
this also can, get another actuations, right? Yeah,
Mhm.
estimating all these kind of structures, maybe, get some more functions and more interesting. I'm thinking that way.
Yes,
Okay.
another project that we are running is we are just adding magnetic nanoparticles inside these fibers, right? So the idea with this is that controlling the density of these magnetic nanoparticles in different sections of the fiber and at different depths, you can actually control the movement
Very
of these artificial snakes, if you allow me the expression, just by passing magnets close to in the neighborhood of the fiber, right? And so we have these little snakes or worms that you can move using these magnets, right?
Yeah, exactly. But this was a super tool, Yeah, we can make more magic stuffs. Yeah.
Exactly.
Yeah, Paul, the possible I can add in more questions. Yeah, because this
Yes,
time,
please.
yeah, it's our first time to have scientists from Latin America. Yeah.
Is that right?
So
Oh,
yeah, that's right. We tried several times. We didn't get through. Now we found the right
well, we
person.
know. We know where to
Yeah,
go.
yeah, we made it. I think, Grissel and Mario exactly was the kind of the Latin American scientist in my mind, like this way, very fast, fashion, passion, with genius ideas like that. So, actually, other people, outside, Latin America, so we are very curious and to see what's going on there. How about the science and technology was in the university or in this place? So yeah, can you guys give us some more ideas?
Yes, well, what I like the most of working in Mexico is working with our people. I think it's the best that we have. We are super proud of our students. I think I don't know if it is cultural, but I in my students I see a lot of creativity, and that there is a problem and they know how to fix it. Even with I don't know, bubble gum, but if there is something that they have to fix, they are not going. We're not going to wait to the perfect solution. We are going to bring some practical solution to keep going. So I think that is the best that we have in Latin America. There are many challenges to overcome in Latin America and Mexico, probably related more to resources and to facilities. But I think in science, that's the way that we think. The most important thing is the ideas and the people. So I don't know, Mario, if you want to add something.
Yes. So I don't know. I will say something a little bit philosophical here. I think that the culture of a place also imparts a particular flavor to the research that you do in that region. I don't know if it is the weather or the food or just the colors that you see or the temperatures through the year. But definitely, I can see that in Latin America, our research is creative, very colorful. You also see that in the presentations that we do, these slides that we use. So it's interesting to see those cultural elements reflected in the way you do research. So it's great to work with Latin Americans, as we said, they are very colorful, very happy people, I will say. So the environment in the lab is nice and friendly, and it's very easy to collaborate. You see that the students collaborate very nicely among them. I will say that we also have challenges. One of the challenges in Latin America is precisely because different cultures can provide different elements to research. We would like to be more diverse in Latin America. So if you go to places like the United States or China or Singapore or England, you see much more diversity in the universities and people coming from different parts of the world. We still don't see that in our country, in our countries. You see now and then people from other places in Latin America, in Mexico, but we will love to see people from China and the United States and Europe coming together and enriching our version of research. That's one challenge. And then the other challenge, of course, is lack of resources, right? As in many of the places in the world, resources are not unlimited. And in Mexico, we are a developing country. So sometimes it's a little bit harder to get resources to continue doing your research. And we have to compensate that with creative ideas, as Grissel said, and bringing more people on board, right, to do things and to become creative. As you can see in our presentation, in Grissel's presentation, we involve a lot of people, a lot of undergrads in our groups. That's a way to enrich and the contributions and to have more mind power in the lab, right? So you gotta use different strategies to be competitive in an international world, right?
Yeah, I like that word. I have been there once, Yeah, I truly love that people's there, very passionate, lovely the things what they are doing, put full of them inside, Yeah, you can feel that. Especially today, Grissel's talk, you can see how excited she is. She's proud of the, that, really enjoy that. So I think that's one part of Latin America, the cultures styles. The people do the things they love, Yeah, they truly put themselves inside. So you see, for the most important thing is a head, right? You have the manpower, the genius, the good ideas. Then you can use all these kind of surrounding materials, right? The research to make some magic. That's how excited we are. So another thing I want to ask is, could you tell us something about the universities or the research labs in Mexico? Yeah, and because we don't have more ideas about that. Yeah.
Yes, well, we work in a very beautiful campus. Tecnologico de Monterrey has different campi in the whole Mexico, and the main campus is in Monterrey. Monterrey is a city in the north of Mexico, very close to Houston, Texas. So more or less you can imagine geographically where we are. And yeah, we have a beautiful campus, very big. We have deer inside the campus and peacocks and dogs and there is very nice people and very nice buildings. And we have several research labs, particularly in one of the research buildings that we work in is Biotechnology Center, FEMSA. That's the name of that building. And Mario was the founder, one of the founders of that building. He was the mastermind behind the design of that building. And I think that building started to propel research a lot in our university. So people started to see us more like a serious institution that made good quality research. And then we had the students. I started to study my master in 2009, and so I had a lab and reactors and incubators and things to make very good quality research. And so that is still there and evolving. Soon we are going to have this new building that is going to be called Expedition. And that is going to be, the concept is to have a place where entrepreneurs and scientists and the community can interact to build new ideas with research that have access to society. So people can have their problems solved by the science and technology that we develop in Tecnologico de Monterrey. So I don't, probably Mario, can you help me to provide some more detail?
You made a wonderful description. Yeah, our university is a private university. It's the largest private university in Mexico. So being private has also some challenges associated. Of course, our students have to pay to be enrolled in the university. We don't receive as much support from the government as we probably should, but that also allows you to be creative as a university. So we have an incredible support from the private sector to provide scholarships for our students. So probably it's one of the universities that provides more scholarships to students. That's the way we are able to attract people that probably do not have, students that do not have the economical resources to pay the tuition, but they are very talented and they still can join us. Grissel studied in this university, the master program and the PhD program with a scholarship from the government. I studied with a scholarship from Tecnologico de Monterrey, my undergrad. So we have to find ways as a university also to bring talent, to enroll talent, and again to try to enhance our educational system with resources coming from different sources. But it's an important university, has been a great player in the economy of the region. As Grissel said, we are in the north of Mexico. There are two very, very competitive universities here in the city. One is private, the Tecnologico de Monterrey. The other is public. Actually, Grissel did her undergrad in the public university of Universidad Autónoma de Nuevo León. That's the name. So it's a nice city that has that flavor of a universitarian university and also an industrial city. So it's great. It's exciting to be here.
Let me follow up a little bit, actually, because you two, I've met many of your students, and you're particularly good at attracting talent. And so how do you do that? Outside of your, creativity and charm and outside of the science, do you have a particular way that you identify people you'd like to join you, and then get them to come and join your groups?
Thank you so much, Paul. Well, we tell them Paul always is our collaborator, and then they come.
Exactly.
Okay, yeah.
Of course,
So,
that
Paul,
helps a bit.
yeah. Maybe this came to my next questions. I think from North America, South America, Latin America, yeah, you collaborate very good, right? So, Paul, you collaborate with different groups, so like Grissel. So, Paul, can you give some, ideas of how you collaborate with these peoples? Yeah.
Yeah. Well, I guess first in terms of identifying people, I met Grissel and Mario and realized how talented and creative they were, and made sure we kept in touch. And we met, I think, almost 10 years ago at a meeting in Cambridge, and spent a lot of time together at that meeting, and I was very impressed with them. When they returned back to their groups in Mexico, we kept touch, and in fact looked for where we could work together. And so not so long ago, two very talented students came up to Los Angeles and really, I would say, cemented that relationship by doing research together with our three groups, and also with Ali Kadamoseni, who was one of the early iCANX speakers, and was the host when Grissel and Mario were visiting faculty at MIT. We also share students and postdocs, so there's this very fluid collaboration between all of us. And then there was a grant that actually supported this collaboration that I came on after it was already funded. That was very kind to pull me in for that. And so we continue to look for ways that we could work together and stay in touch. And even through COVID, we're able to keep all that up. And now that the world is reopening, we're looking for other opportunities to spend time together.
Oh, wow.
And actually, I have an idea based on the talk that I'll reach out to you afterwards about, that we should certainly try. But maybe better to write it down first and make sure to work.
Yeah. So, Paul, we're proud to say that today we have 21,000 audience online, and a lot of people with feedback very good. They say that's the future. That's the future for this field. Yeah.
Yes.
So, yeah, proud of you. Yeah.
Fantastic. Well, thank you so much for this wonderful talk and discussion. Can't wait to see you in person somewhere in the world soon. I'm not sure where that'll be, but we'll look forward to having some time, and let's put together a call so we can talk about what the next steps in research are. And so I think we have a certificate here. I'm waiting for the slide to change. There we go. So thank you so much for giving this iCANX talk. When you visit Beijing, you'll see the wall on which you'll be immortalized in this beautiful building that Alice has set up there as the iCANX headquarters. And actually, this also pulls you in as a permanent member of the iCANX family. So congratulations on this very beautiful work, and for joining the family. Thank you so much.
Thank you so much, guys, for everything and for the invitation. And I really enjoyed this panel. You made my month.
Yes,
Great.
so now you are most famous, Latin American scientist in the world. Oh, yeah. Okay, yeah. Oh, this was
very nice. So please stay tuned in the coming weeks. We have a terrific program, including next week. with Lan Yang as our speaker, and she'll be hosted by Professor Lan Fu from Australia, where I'll be heading actually in 3 weeks.
Yes. Okay. That's
Hey, thank you once again, and we'll say a good morning, good afternoon, good evening from all of us at iCANX. Thank you very much.
Thank you.
不再是奇迹，不再是幻想，此刻真感觉全世界为我鼓掌。不必太在意身旁惊奇的目光，可以点点头，可以放声歌唱。我创造奇迹，我拥有梦想，我希望看见所有骄傲的脸庞。不再为曾经失败放弃或感伤，努力才是真的方向。I can， 没有什么可以阻挡心中无限的力量。I can， 你也能够像我一样飞越最高山岗。I can。没有什么可以阻挡心中无限的力量。I can， 你也能够像我一样飞越最高山岗。创造奇迹，我拥有梦想，我希望看见所有骄傲的脸庞，不再为曾经失败放弃或感伤，努力才是真的方向。I can， 没有什么可以阻挡心中无限的力量。I can。能够像我一样飞越最高山岗。I can， 没有什么可以阻挡心中无限的力量。I can， 你也能够像我一样飞越最高山岗。能够像我一样飞越最高山岗。
祝你生日快乐！该吹蜡烛了，
来，我帮你。
祝你生日快乐！
生日快乐！
好漂亮的花，我很喜欢。
这是我的礼物，多层陶瓷电容器。
哇，这是多层陶瓷电容器吗？
失败了。